Hi, I'm Fumika UENOSONO. My research theme is effect of bitter and sweet receptor stimulation on ciliary movement in human bronchial epithelial cells. First, let me introduce the background of this research. In our airways, tiny hair-like structure called cilia help remove harmful particles and pathogens trapped in mucus. Therefore, maintaining normal ciliary movement is crucial preventing for airway diseases. Previous study had found that beta receptors are expressed on the cilia and bronchial epithelial cells, and that beta compound activates the ciliary movement via increased intracellular calcium. Figure 1 shows the signal transduction pathway in human bronchial cells. First, beta compound binds to the receptor. Second, the beta-gamma dimer of trimeric G protein activate phospholipase C beta-2 and convert membrane lipid PIP2 to 1,4,5-inositol triphosphate IP3. IP3 binds to the IP3 receptor. Calcium is released from ER and the intracellular calcium increases. Fourth, calcium channels open and calcium flows in from outside the cells. An increased intracellular calcium and subsequent activation of protein kinase might phosphorylate axonal dyning and activate ciliary movement. Now, I will talk about the methods. Firstly, NHB cell culture. NHB cells were cultured in flasks and seeded onto transwell. The apical surface of monolayered cell was exposed to air for more than 21 days to differentiate the ciliated cells. This method is called ALI culture. Figure 4 shows the differentiation process of NHB cells. Red is an anti-acetyltubulin antibodies, a marker for cilia, and blue is DAPI, a marker for anti-cell nuclei. Each result contains approximately 5 cells. Cilia is fully differentiated by the 21st day. Secondly, fluorescence antibody staining, IF. IF was performed on NHB cells cultured for 21 days of ALI using an anti-T1R2 antibody, an anti-acetyltubulin antibody. Finally, observation of ciliary movement. The filter was stained ciliated cell was set into observation chamber and the solution on the apical side was perfused. The ciliary movement was observed and recorded by an inverted microscope connected to a high-speed camera. Ciliary beat frequency CBF was calculated the number of waves in 1 second. Procaterol, an adrenergic beta-2 receptor agonist, was a positive control for ciliary movement. I will talk about results. Please look at figure 6. Sweet receptors were detected with anti-T1R2 antibodies in green, and cilia were detected with anti-acetyltubulin antibodies in red. The two results were combined, and the overlapping area were detected in yellow. Z stalk has an apical side on the right side, a basal side on the left side, and above the apical cell membrane was stained yellow. Therefore, T1R2 sweet receptors are expressed on the cilia and apical cell membrane of NHB cells. Figure 7 shows the effect of procaterol 10 nanomolar and denatonin 1 millimolar on ciliary movement. Procaterol is an adrenergic beta-2 receptor agonist, and denatonin is a beta receptor agonist. In graph A, the vertical axis shows the CBF ratio, and the horizontal axis shows the time. The black line shows the perfusion result with controlled solution. The orange line shows the result with procaterol. The blue line shows the result with denatonin. Graph A shows the CBF value don't change during controlled perfusion, but reversibly increase during procaterol and denatonin perfusion. In graph B, the vertical axis is CBF ratio. When the control is set to 1, it represents the maximum rate of change in CBF during denatonin and procaterol perfusion. The number in parentheses indicates the rate of increase percent in CBF values. Graph B shows that denatonin and procaterol increased CBF to a similar extent. Therefore, denatonin, a beta compound, significantly increased CBF. Figure 8 shows the effect of sweet compounds on ciliary movement. I used 4 types of sweet compounds: aspartame, sucrose, saccharin, and sucralose. Aspartame, saccharin, and sucralose are synthetic sweeteners. The number in parentheses indicates the rate of increase percent in CBF values. Aspartame significantly increased CBF at 3 millimolar and 5 millimolar. Sucralose significantly increased CBF at 50 millimolar and 100 millimolar. Saccharin significantly increased CBF at 0.1 millimolar. Sucralose significantly increased CBF at 1 millimolar. Consequently, aspartame, sucrose, saccharin, and sucralose sweet compounds significantly increased CBF. To determine whether sweet receptors mediated the increase in CBF, I used Lactazole, a sweet receptor blocker. Figure 9 shows that Lactazole didn't increase CBF, and the aspartame-induced increase in CBF was completely inhibited by co-perfusion of aspartame and Lactazole. It indicates that the increase in CBF caused by aspartame is mediated by sweet receptors. To determine whether an increase in intracellular calcium mediated the increase in CBF, I used BAPTA-AM, an intracellular calcium chelator. Figure 10 shows the pretreatment with BAPTA-AM didn't increase CBF, and the aspartame-induced increase in CBF was completely inhibited by pretreatment with BAPTA-AM. Therefore, it indicates that the increase in CBF caused by aspartame is mediated by an increase in intracellular calcium. Finally, I will talk about discussion and conclusions. Beta receptor expressed on the cilia of bronchial epithelial cells are thought to act as a defense mechanism by preventing harmful substances and activating ciliary movement to expel them. The psychological function of sweet receptors expressed in NHB cells is unclear. They may detect sugar and activate ciliary movement to prevent the proliferation of microorganisms by removing excess sugar from airway mucus. In conclusion, sweet receptors are expressed on the cilia and apical cell membrane of NHB cells. Treatment of NHB cells with bitter and sweet compounds seems to activate ciliary movement via increased intracellular calcium. Thank you for listening.